My proudest accomplishment is the amount of research I've been able to do with undergraduates here. It feels so valuable to get to work with undergraduates. Like I benefit from it, but also I think that they benefit from getting to camp for the first time, getting to see turtles in the wild, for many of them for the first time. I've had students who've never left Chicago until doing fieldwork with me. And so every summer, just getting to share with them the beauty of what Illinois has to offer, what Wisconsin has to offer, how easy it can be to go camping and like break through that barrier is the thing I'm most proud of. That I get to do that every summer, take students and just change their lives means a lot to me.
My guest today is Dr. Beth Reinke, Associate Professor of Biology here at Northeastern Illinois University. Beth, thank you for joining me.
Thanks for having me, Paul. I'm happy to be here.
So one of the things you study is coloration and the evolution of coloration.
Mm-hmm.
Could you talk about that a little bit? Introduce us to your work and some of the species that you study coloration in.
Sure. So coloration is a really fascinating topic, not only because it's aesthetically pleasing and easy to use for science communication, but it also is really interesting. It affects every facet of an organism's life. Their behavior, their environmental context, their evolution, all of these things impact and are impacted by their coloration. And so part of the reason I really love studying it, not only is it easy to communicate and talk about, but there's so many different questions and directions you can go with studying color. There's so many animals that have coloration we don't fully understand. There's so many nuances to colors and patterns in the animal kingdom that I'm never going to run out of things to study, and I really appreciate that. However, most of my career I've spent working on a single species, trying to answer one single question, which is why are painted turtles painted? So I've spent a long time working with that particular species, specifically kind of the western range of that species, which many populations have really bright orange ventral coloration that we don't understand the function of. And that's what a lot of my work has been around, trying to understand that.
Do we have a theory as to why the painted turtles are colored in that way?
Yes. So I spent a lot of my PhD work kind of ruling out hypotheses. And have landed on a potential hypothesis right now. I currently have a National Science Foundation grant to be investigating that. And I may have an answer or two in the next couple years. We have currently 300 hatchlings in the lab. We're overwintering them and doing some work with their coloration to try to address our working hypothesis, which I don't know. Do you want the full story or do you want?
Let's do the full story.
Okay, so generally, animals have bright color. When animals have bright coloration, it's often a signal, which means it's trying to communicate some sort of information to other animals. And we can loosely kind of divide that into conspecific signaling, so signals that are meant for other individuals of the same species, or heterospecific signaling, signals that are meant for predators or other individuals of a different species. So you can think about things like warning coloration or even camouflage, we can kind of consider in that heterospecific camp, versus signals that are used for mate choice or communication to other individuals of the same species or in that conspecific camp. So a lot of my early work was trying to figure out, is this orange coloration a signal? And if so, who is it meant for, right? Usually when you have a bright coloration, it has a function because it's going to make the animal more conspicuous. So one of the first things I did was visual modeling, which tests essentially it removes the human bias of vision and lets us quantitatively answer questions about how a color looks to another species. So you take into account information like the wavelengths of light that are bouncing off the object of interest, the light environment, various aspects of the physiology of the viewer's eye. So we did this for a lot of the common painted turtle predators and found that to all of them, the orange color of the plastron, which is the name for the ventral shell, is really conspicuous. It stands out against most of the common backgrounds where it would be seen as it looks to us. It's also really conspicuous to us, and it's conspicuous to their predators. So that tells us, okay, this isn't for camouflage, but maybe it's meant to deter predation in some way. Maybe it's a warning coloration. So we did predation experiments with models of turtles, turtle hatchlings specifically, just because that's when they're most often predated. And we found that the ones that we painted in orange color that match natural hatchlings were not attacked any more often than the ones that were kind of a more dull tan that reflects other turtle species, plastrons. So it didn't seem to be deterring predation at all, and it's super obvious to predators. So we also did some work on just looking at, could this be a signal to other painted turtles, we don't see any behavior that shows off that ventral side of the shell. It's really weird because, well, if you picture a turtle and it's swimming, its belly is facing downwards. So you'd really, if you're gonna try to show off that shell, you would be having to do some sort of behavior that really flashes it. And we haven't seen that. We also see no evidence of sexual dichromatism. So males and females don't have different colored orange, different colors on their shells. And because of those reasons, we can kind of rule out a lot of these conspecific signals. So I did several experiments to test each of these things, kind of was just knocking hypotheses off the table. Doesn't seem like the colors for this or for that or for that, until basically I had no more hypotheses for signaling left. So if the painted turtle has this bright orange color, and presumably it has a function, although that is a presumption that we're making, what do we do next when we run out of these hypotheses, right? So one of the things I did was try to figure out, where is the color coming from? How is it made, right? In animals, color is created one of three ways, either with a pigment, a structural color, like the nanostructures of the actual tissue that are changing the light's refraction, or a combination of those two things. And what we found is that the pigment, it is a pigment that's creating the color, and it's a carotenoid pigment. And this is super cool because carotenoids can't be synthesized by any vertebrate. And so they have to be getting that pigment from their diet or from maternal yolk or whatever, which means that there's probably a cost to it. It means they're having to obtain it, they absorb it, metabolize it, and express it in tissues. So they're probably not doing it for no reason, or you would expect that to evolve out eventually, right? So that tells us there probably is a functional significance. It's probably an adaptive color, but if it has nothing to do with signaling, then what in the world is it for? And so this is where I kind of had to become a physiologist and start learning about molecular biology and how carotenoids and other pigments can work within a body. So essentially, many pigments, including carotenoids, can be antioxidants, which is really important for organisms. Antioxidants are really important for organisms because reactive oxygen species are these unstable molecules that get produced as a natural byproduct of living, of everything. And you have to have antioxidants to combat the ROS, the reactive oxygen species. If you have too many ROS relative to antioxidants, you get oxidative stress, which eventually, which causes organismal stress, cellular death. It can eventually cause organismal death if you have too much of it. Now, why that might be relevant for painted turtles is that painted turtles have a really unique overwintering behavior. As adults, what most freshwater turtles do in the Midwest, if you picture, we've got lakes and ponds around here, in the winter, they freeze over. So the organisms in the water have to be able to get air somehow. Painted turtles are ectothermic, or all turtles are ectothermic, meaning that their metabolism and oxygen demand, heart rate, all of these things decline in the winter. And so most species of freshwater turtle can just hang out in the water column and absorb enough dissolved oxygen to get by in the winter. Painted turtles, however, don't just hang out in the water column. They go down into the muck underneath the water into anoxic conditions. So painted turtles can survive complete anoxia for months over winter, which is very strange and has been studied a lot. How are they able to do that? How are they able to go without oxygen? Relatedly, the hatchlings also have a really weird overwintering behavior. So in many places where turtles, including painted turtles, lay eggs, they do so in early summer, in May and June. The hatchlings hatch in July and August. They climb out of the nest, they go to the water, live their life. Painted turtles, especially in the northern part of their range, will hatch out of the egg, but stay underground. And they'll stay underground for 6 or 7 months until April or May. So they're going months out of their shell, but still in the nest, never seeing sun, never eating, never drinking. And they can actually survive complete somatic freezing. So they're freeze tolerant. So freezing and anoxia are both crazy behaviors that require a lot of similar adaptations because you have to be able to survive oxygen being cut off to all your tissues for a period of time. And what happens when oxygen is reintroduced to all these tissues is what's called reperfusion injury. So reperfusion is the process of oxygenated blood being reintroduced to tissues. And essentially the body kind of kicks into overdrive and produces way more ROS than usual. So you'd expect organisms that have to deal with this type of reperfusion injury to have a lot of oxidative stress. And yet we don't see that in painted turtles. So my current working hypothesis, this is not published yet, we're still working on it, is that those carotenoids might be stored in the shell relatively inertly until winter, where they may be moved to areas where they're needed to combat oxidative stress and the ROS that are produced during the thawing process. One thing that's really cool about this species is that the DoD and other organizations were really interested in understanding how are turtles able to survive this in like the 90s. So they funded a ton of research into it. So we know a lot about the physiology of overwintering, but we don't know a lot about the physiology of the thawing process, of that like recovery process. Most of it's looked at how are they able to survive the ice formation and all these other things. So currently I have hatchling turtles in the lab who were given different levels of carotenoids, and we'll be overwintering them and freezing them and measuring oxidative stress before and after and seeing if the ones with more carotenoids and brighter colors or more carotenoid rich colors are the ones that have lower oxidative stress or not. Because if they do, then that gives us a huge clue that the painted turtles are painted to survive winter. So
the hatchlings don't eat anything over winter then?
supposedly, we're finding in the lab they do seem to eat our paper towels and the substrate that we have there, but they're excreting it right out. They need to excrete everything so that ice doesn't nucleate around any objects in their stomach. So if they're eating things, it seems to be nutrient poor, and they'll still.
And the hatchlings already have the coloration.
Yes, they do, exactly. And actually, we see bright plastron colors in many of the emidid turtles, which are the turtles in this particular family the painted turtles are in, but usually they fade over the first year. And so you'll see often the yolk sac is like right in the middle of the plastron, and you can even see color emanating out of that yolk sac sometimes. The reason yolks are yellow is because of carotenoids, so it makes a lot of sense. But then you see that color disappear in most emidid species. Painted turtles are one of the exceptions, where they keep this bright orange color throughout their life, even as adults. In fact, we don't see a consistent change in color with age in this species at all, which is fascinating.
And you'll see different patterns on different painted turtles, right?
Yes.
So is that just due to the amount of carotenoids that an individual has?
So great question. So the patterns actually may be a separate issue entirely. So the way a painted turtle plastron looks is it's bright orange in the western area where we work on them. They're bright orange, and they have a black, what we call a plastral pattern on top. And that black plastral pattern is melanin based. So it's melanins layered on top of the carotenoids. So we've done another study that we'll be submitting soon, looking at whether the plastron pattern has a role in this anti-predatory strategy of maybe when the hatchlings are trying to get to land, maybe breaking up the background or just making it harder for a predator to see where the animal is. And we actually did find that painted turtle hatchlings with more complex plastral patterns were 10 times less likely to get attacked than ones with just a simple black line. So it does seem like the plastral pattern that's on top of the orange carotenoid-based pigment might have an anti-predatory role. And it could be shielding a lot of that orange carotenoids, which kind of makes sense. If they're really just storing it kind of for the physiology, for the physiological function, it makes sense that they'd try to be masking it with this melanin if they need to.
Yeah. But then why wouldn't they mask all of it, right?
Exactly. Why wouldn't they mask all of it? Why do we see so much variation in wild populations? Like there are hatchlings with a really simple plastral pattern and adults. So why are we seeing such variation if this is really so important? And maybe it's not. Maybe that's what it comes down to, is that it does help them, but not so much that the simple ones are kicked out of the gene pool. Don't know.
Because within adults, then the coloration, there's no association with predation in coloration, right?
Yeah, not that we know of. We're not seeing that in adults at all.
But in hatchlings, we are.
Just with the plastral pattern, not the color.
Just the pattern.
Yeah, just the complexity of that pattern shape. Yeah. Yeah, it's really strange.
Yeah.
I just had a student work on this summer where we finally have long-term enough data. We've been studying the same populations, one population for 30-something years, but we've only been working on the color for 5. And another population, the one in Wisconsin, I've been studying for 16 years, and we've got color data for about 10 of those years. And so we're finally able to get really a good look at how a particular individual's color changes as they age. And what we're seeing is that their pattern doesn't change at all. It's, maybe slight distortions, but the pattern stays the same throughout their life from juvenile to adult and beyond. And the color seems to change almost yearly. We're seeing it go up and down and up and down. It's not a consistent pattern at all.
When you say color, you mean the vibrance
or
the
orangeness?
actual
Yeah, the orangeness. Yeah. So I could send you a picture that you can put up too of just like all the plastrons of one population in 1 year, and you see how much variation there is even in one population in 1 year, let alone from one individual from 1 year to the next. So we're seeing a ton of variation in how much carotenoid is there. Now, could it just be a result of like these kind of random stochastic changes in diet or availability of carotenoids in the environment? Yes, but you'd expect to see it more consistently across the population, where we're seeing variation in individuals. So it could have something to do with how well an individual can metabolize carotenoids, how many they use to mitigate oxidative stress that spring before. We really don't know.
Why wouldn't we see it on the dorsal side?
Presumably, evolutionarily, maybe it's on the ventral side just because that's where it's hidden. And so the dorsal side is more obvious to predators. If you imagine a turtle swimming and you're a raccoon or a fox or something looking down into the water, they have that dark color that is typical of animals where they're counter shaded. So they have a dark dorsal side and a light ventral side. So if you're looking down or looking up, you'll blend into the environment. So maybe the orange is just on the ventral side because that's where it's more cryptic, even though when you see it, it's not cryptic at all. But if it's facing down, you might not see it.
And what is the average lifespan of a painted turtle?
In the wild, 30 to 50 years.
30 to 50
They
years.
live a long time. Yeah. So we've done some really fun work on aging and longevity. That's one of the benefits of doing this type of long-term work, is that then you can get actual accurate survival estimates from year to year. And we found that they live a pretty long time in the wild.
So you said there's been a lot of research done on how they overwinter or survive these freezing conditions. So what explains this enormous phenotypic variation or color variation that we find throughout the animal kingdom?
That's a big question, Paul.
Yeah.
What explains it? I don't know. Environmental heterogeneity, evolutionary stochasticity. I don't know. So much. There's variation everywhere, right? Mm-hmm. Of course, we oversimplify evolution of thinking about how it's going to lead to organisms being a perfect fit for the perfect environment. That's obviously not true. That's an oversimplification, but it is a striving towards. We see these processes where individuals that are better adapted for their environment are going to pass on their genes, which means those are the ones that are just the way evolution works. And so you can expect, with as much variation as we have in environments, in ecosystems, in the context in which an organism exists, we're going to see an evolution of diversity, where we're going to see a lot of different colors and patterns and variation across the animal kingdom. So the answer is a lot of things explain.
Right. But is there any data to support greater color diversification as an anti-predatory response, or for camouflage, or do we have any data along those lines?
I think it's really context dependent. Like, so picture a bright red feeder shrimp, right? It's bright red. To us, that looks like a warning coloration. But when you imagine it in its native habitat of a coral reef, it's cryptic. It's blended in, right? And so I don't think that we can explain color diversity as easy as just saying, oh, most coloration is an anti-predatory function, or most is cryptic. I think it's so entirely environmental dependent, but that's part of what makes it so fun to study, right? Is that you're never just studying the organism. You have to think about the population, the ecosystem, the interactions it has with other organisms, and this whole kind of context thing.
Because coloration, we'd assume through evolution, would have some evolutionary explanation, right? There would not be a situation where it wouldn't have any functional purpose.
That's not necessarily true. So we do see what's called non-adaptive coloration, which is thought to be just kind of a baseline color. So picture, like, think of an example. Melanin-derived colors are often the ones that are considered to be the non-adaptive, right? This is just a thing that the body produces really easily. It doesn't make the animal survive better, but it doesn't make it survive any worse either. And so having coloration in a particular hidden spot that's black or brown or whatever doesn't necessarily mean anything. There's a whole, adaptationist paradigm where you could think about how do evolutionary biologists approach questions about the functional significance of traits. But generally, when we're asking questions about color, we want to be thinking about, is this likely to be an adaptive color? Because if not, then it's probably not worth investigating, because it's not going to significantly impact that organism's life history. Don't know if that answered your question, but
no, it
You're asking me big questions here.
Um, what other species are you looking at in addition to the painted turtles?
So I'm doing some work right now on squamates in general, which is lizards and snakes, and we're kind of looking at how the evolution of color, like this is a macroevolutionary approach, how the evolution of color and sexual dichromatism might correlate with things like parental investment. So that's a really fun, like, large scale approach, thinking about how these things might have evolved together. I've done work previously on butterflies in the past that we haven't published yet, but looking at similar things, sexual dichromatism, how male and female differences have evolved from a macroevolutionary perspective. Um, and then lately, I keep working on birds, although I'm not doing so much bird coloration as more bird preservation work. Here at NEIU, we have a lot of glass corridors that were killing a lot of birds. And I was assigned to teach local fauna and ornithology, and so I thought, why let these go to waste? Started collecting the carcasses with permits, of course, and now we've been doing work to preserve them and kind of practice some of those techniques to keep study skins and then taxidermy. So that's a different direction, not really working with coloration at all, but we have these specimens now so that data like coloration can be collected from them. And that's one of the nice things about having a collection, having a natural history collection, is you can collect data years later when a project occurs to you or whatever. So it's a huge asset, especially at a small school like ours, to have a natural history collection that we can get data from as needed in the future. Maybe before and after data, you just never know. So
What are some of the findings that you've found within butterflies? Like,
we were comparing rates of evolution of the dorsal side versus the ventral side. So working with turtles, where I'm focusing pretty much exclusively on the ventral side, got me really interested in thinking about, okay, what about these hidden areas of color that, hidden may or may not be actually hidden. And butterflies are a really cool example because they're two-dimensional, essentially. We can break down their wings as being this flat surface, where one surface is visible when they're flying, the dorsal surface, and the ventral surface is visible when they're resting. And so you might make hypotheses about, okay, I would guess that the ventral surface probably has more to do with camouflage, where the dorsal surface might be more to do with signaling. And if that's true, how do these sometimes competing selective forces act on the same structure that technically has two sides? And so you can look at, with a macroevolutionary approach, you can look at how the relative strengths of natural and sexual selection, for instance, on different color traits across those groups. So I don't remember what we found, but that's what we were looking at. Sorry.
well, going back to the painted turtles, have you found there's no difference or correlation between male and female coloration and reproductive success?
Nope. So we don't see, so firstly, males and females don't differ in color at all, which is really strange. If you look at a box plot, it's just, they're pretty much exactly equally variant. In terms of carotenoid chroma, ultraviolet reflectance, all these kind of objective measures of coloration that we can do. We have looked at things like, could this color be an honest signal? So honest signals are colors or ornaments that convey some sort of information to potential mates. So you'd expect that information to be related to something like the health of the individual, for instance. So one of the studies we did a few years ago with an undergraduate, Jonathan Stasiak, is we looked at is the orangeness of the plastron correlated with the bactericidal capacity of the blood, right? So are they advertising something about how good their immune function is? And we found no correlation in two populations, Illinois and Wisconsin. What's interesting is we also look at the carotenoid chroma, or the yellowness of the neck stripes, because in many turtle species, those neck stripes are signals. We know in related species like Trachomys scripta, the red-eared slider, they have like a red ear patch where the redness is correlated with immune function, and it seems to be sexually selected. So we found that in one of our populations, the carotenoid chroma of the neck stripes isn't correlated with immune function at all, but in the other, it is. And so that was really surprising, because generally when we're talking about things like honest signals, we think about it at a species level of like, this is what's happening to the species. And that's in fact not what we found. It's what's happening at that population level. And we don't know which population is the weird one, right? Because we looked at two. And so it's could be that most painted turtles have this honest signal in their neck stripes, and it could be that most painted turtles don't use that honest signal, and this population is the weird one that ended up with it. So it just kind of comes back to this theme of variation and how much there actually is in the animal kingdom and how little we understand about the variation in color, the variation in function, and the diversity.
Are painted turtles sexually dimorphic?
They are. Females are much bigger. The males also have elongated foreclaws that they use for courtship. They wiggle their claws in the female's face, and their tails are different lengths for mating. So, yeah.
Mm. And is anyone looking at this dancing that they?
I don't know if anyone's doing it right now. It's hard to get them to do it in captivity, but there are videos, like you can just even Google like YouTube painted turtle mating or whatever, and you'll get the courtship, because it is really funny to watch. And when people keep pet turtles, sometimes they see it. It's actually occurs across the family, so it's not just the painted turtles that do it. But I don't know if anyone's looking at. I know people have looked at like the length of the claw and like whether that's what matters, and it's kind of ambiguous results. So
no one knows what the females are selecting for necessarily.
Not really.
Can you share maybe a moment from a failed experiment that led to a surprising kind of finding or led you in a different direction?
Well, I can certainly share failed experiments, yes. So when I was in grad school, I had ruled out all these hypotheses for why painted turtles are painted. I had this idea about maybe the carotenoids are antioxidants that are important. So we collected eggs, and that's easier said than done, at least at the population I was working with at the time. This was in northern Wisconsin. The turtles we work on are in a single recreational lake that's surrounded by like wetlands and private property. And the turtles come up to nest, but it's not all in one place. They're really pretty disparate. And so oftentimes we would spend our afternoons just driving the roads looking for turtles that are trying to cross the road, because they're usually looking for nest sites. We could grab them, we'd inject them with oxytocin, and then wait for them to lay the eggs. Sometimes that waiting took 24 hours. I don't know why. Maybe we were using the wrong dosage or something, but it was taking forever. And so that meant checking on these turtles who were in these bins with water every 15 minutes for 24-hour periods. So it was a lot of work to get these eggs. So we'd get the eggs, then put them in vermiculite, which is what we incubate them in. And I was trying to inject them with different levels of carotenoids. I was also doing a removal group, so we were trying to remove some carotenoids from the yolk to see what happens. So it's a ton of time, of sweat, of tears, of just not sleeping, just watching these turtles for several weeks at a time, just to get a few hundred eggs. Brought them all. I was at Dartmouth at the time in New Hampshire. I was catching turtles in Wisconsin, so then I was driving them back, trying to keep them at a constant temperature the whole time. Anyway, you get the idea. It's a lot of work. That first year, 0 of them hatched. Every single one did not make it. Didn't know why. So next year, tried it again. Got them all back. 2 hatched out of 200 something. You can imagine, it's really stressful. I can only do this once a year,
Yeah.
Horrible. So I ended up doing a correlational study. I got a bunch of hatchlings from commercially, just measured their coloration, put them through this overwintering, and we did find that the color changes over the winter during the freezing time, and that the oxidative stress of the brighter ones was slightly lower than the ones that were less bright. So that suggests to us, okay, they're moving the pigments, which means they're probably using them, unless it's some sort of weird passive movement. And we do see variation in how much oxidative stress the hatchlings have. But it's not an experiment, right? We didn't manipulate the carotenoids to see if that's what does it. So when I got this NSF grant 2 years ago, I decided to redo these experiments. Now I'm closer to Wisconsin. It's way less driving. So I did it last year. This population, we actually did it from the Illinois population that I've been working with now, which is way easier. We stay, our field crew stays on a public campground, and the public campground is the nesting ground. So the turtles are literally coming up around you. It's not, hours of labor to find a single nesting female. They're everywhere. You're trying to keep track of all the nesting females around you, and they just lay their eggs, and then we can get the eggs back, excavate them. So none of the waiting, none of the lack of sleep. Anyway, way better system to get eggs. So I injected them with the different levels of carotenoids, brought them back to the lab. I think I had 30 of the 300 hatch. So failed again. So it's the injections that are the problem. So this year, we, instead of doing injections, we topically applied the carotenoids, and it was indeed absorbed into the egg, and we were able to verify that with some sacrificed eggs. And so it's a huge relief. 4th time's the charm. All this blood, sweat, and tears went into trying to collect these eggs and figure out a method that works to actually manipulate the pigments. And I think we have it now. So I'm very happy. The turtles are alive, doing well. We had everybody hatch except I think we had like a 95% success rate, which is very normal. And so fingers crossed it keeps going well and that.
How did you find or select your field site in Wisconsin?
Oh, so in Wisconsin, my grandparents had a cabin up there that I spent my summers at when I was a kid. And I would catch turtles there just for fun. And so when I was an undergrad, I had a really great mentor who was a graduate student. And I met her because I wanted to get involved in research, and she was doing a project with snakes. So I asked to work with her, and we were working with, there were dead snakes. It was like a morphometric study. But it was really fun, and I got to know what research was as an undergrad. And she encouraged me to try to do my own research project, which is very overwhelming when you're an undergrad. You have no idea what that even means or what that entails. So she asked, well, you want to work with animals, you want to work outside. Where is a place where where their animals are? It's like, well, my grandparents, I catch turtles there for fun all the time. And so she encouraged me, she helped me write a grant proposal to the local lake association up there, who for some reason volunteered to support my project. So I kind of cold emailed them and they said, sure, yeah, here, have some money to buy this research equipment. And I started that study. And that's the same study I'm running now. So 16 years later, I go every summer, the same 2 weeks of the year. We catch turtles, we individually mark them, we collect data, collect coloration data, but also morphometric data, and we're able to monitor the population that way too. So,
have we noticed any change in due to climate change with their hatchling window, or hatching window, I should say?
So it's a little harder to say up there because we're only there during the nesting season, and the nesting season has been shifting, but it's really erratic. We've had some like really cold springs, we've had really warm springs, and it seems to shift accordingly. The problem with being so far up, we're like way in northern Wisconsin, it's such a short growing season. So there's a small window of time for those turtles to be active, and they seem to be making the most of it. But we're only there for the 2 weeks during nesting. So we don't know if that hatching is shifting. It's really hard to tell. But we are seeing the nesting shift back and forth.
So do you have like a scout up there to tell you when? Yeah.
Yes, I do. Yep.
That's
Says I saw a turtle on the road and I say, let's go. So, yep.
Pieces of equipment that you use in the field.
Yeah, it's super variable. So at the Wisconsin site I was just talking about, we catch the turtles almost entirely from kayaks. It's really fun. So we're in these like super eutrophic bays of this lake, lots of duckweed and lily pads everywhere, and you really have to develop the search image for what a turtle head looks like. And then we use dip nets. So we're literally just paddling. You see a turtle, you scoop and try to grab it. And we can get a few hundred in a day that way. It's such a dense population. In the Illinois population, so the Illinois population was started by Fred Jansen, who was at Iowa State at the time. He started it for his PhD, I think, 35 something years ago. And he was mostly studying nesting females. And then I was a postdoc at Penn State working with him and some other people. And they wanted to start getting more population level data, so they were trapping males too. And then he moved to Michigan State and wasn't going to maintain that field site, so I took it over 5 years ago. So he still does work there though. So we both work together to do it. But at that site, we can't catch turtles from kayaks. It's just not, it doesn't work. Don't know why. They're less dense, like for sure. The environment's a little bit different, but they're also way more skittish and we just aren't able to get them that way. So there we're getting the females that are coming up literally to the campground where we're staying. And then we trap males and females with really big hoop nets. They're like these funnel traps that we set in the water with a fike that connects them. And so a turtle will be swimming along, it hits the net, and it has to go left or right, and then it'll eventually swim into the funnel of a funnel trap. So for that site in Illinois, we're using, I say Illinois, it's on the Mississippi River, so it's Western Illinois. But we're using waders and traps, and that's primarily how we're catching them. And then Wisconsin, it's entirely dip nets and kayaks. So really different experiences for the same species.
Yeah. And then are you tagging any of the turtles then? And
yes.
Seeing them year after year after year?
Yes. So every individual we catch is marked. We also do that differently. And that's partially a result of inheriting the site that Fred Jansen started, where he was using a notching system. So they use like files to mark a unique ID on the side of the turtles. In Wisconsin, my very first year, I tried to do that and the turtles bled. And I said, oh my gosh, how is everyone doing this? Like, why would you do that when it's so horrible? And so I stopped doing it. So I microchipped them. It's only recently that we realized, wow, wait, it's really weird that those turtles bled and the ones in Illinois do not at all. Like, something's actually functionally different with the shell morphology. And it wasn't until I had a student a few years ago who was collecting samples with like a nail file from both the populations that we realized how incredibly different their shell anatomy is in these two sites. And we don't know why yet. So that's something I'm hoping someone will want to work on in the future.
Yeah, right.
They're way thicker in Illinois, and I think that's more typical because many people use this notching technique and it's fine. But the Wisconsin site, the turtles are so sleek and the shells are very thin, clearly highly vascularized, and we just don't know why they'd be so different. And so there we're microchipping them. Yeah, it's very weird.
So we think could there be something because it's further north?
It could be
climate.
Yeah, it could be just they have a shorter growing season, and so they're just putting less energy into the bone density. It could be maybe that there's a release from predation. I don't even think that makes sense. I think they have, if anything, some more predators up north, bigger predators. Or it could be just that maybe this is all tied together and it has something to do with being able to mobilize that pigment. Because even though in the Illinois population they still are freeze tolerant, it's a less severe winter than in northern Wisconsin. So maybe it has something to do with the color after all.
Yeah. And then have you created a database of the
Yes, of the plastron patterns. Yep. We've got, in fact, one of the things we're doing with this grant is working with a software company that's helping us kind of automate a way to analyze the pattern and quantify the patterns. I'm also partnered with a geneticist here at NEIU who's helping us try to identify just factors related to coloration in the genes and the pattern. So we're really trying to get at the heart of like, how is this pattern formed? How is it varied? We see that there's a lot of similarities in siblings. And we got that just from hatching these turtles in the lab. We see that plastron patterns are really, really similar among siblings. So how much can we get about heritability, and how can we use things like neural networks and AI to get estimates of like relatedness just from looking at the plastron patterns? So that's kind of the direction we're headed right now, which is really fun to just explore what can we learn about where an individual came from just based on its plastron.
And do we notice any pattern differences between the Wisconsin population from the Illinois population?
Yes. Yeah, and it's interesting because the actual patterns are probably not different, but in the Illinois population, because of being on the Mississippi River, they have this buildup that completely obscures the plastrons. So occasionally we'll get individuals that don't have it at all, or individuals that just shed some scutes. And so you can actually see the underlying pattern, but mostly it's this dark red that covers everything. It's like a dark red brown. It's not a bright color at all. It's like really a muddy brown color. So that's what we see in the Illinois population, where the Wisconsin one is vibrant, bright orange.
And when you say buildup, what do you mean by buildup? Is that? It's
literally on top of the tissue levels. Like, we don't know. You can kind of scrape it off with your finger. It's coming from the environment. Some publications have called it iron oxide. Some have called it tannins, just from the color of the water. We really don't know what it is. I need to get a chemist involved, essentially, to analyze it and figure out what's happening here. But what that means is that physiologically, it might not affect anything. If that color is, only has a physiological function, then having an external covering might even be helpful. But if it has any sort of signaling or other things that we haven't investigated, then we'd expect to see these differences between the two populations. So makes it kind of a nice study system.
Yeah, definitely. One of your other interests is this melding of biology with art. Could you talk a little bit about that?
Yeah, sure. So my best friend is an artist, Caitlin Patton. She's based here in Chicago. And she was one of the first people who helped me with this turtle study when I was an undergrad, and we were just trying to figure out what it is. She came up with me to Wisconsin. She helped me caught turtles. And initially, the study was very population ecology focused. We were looking at population growth rate across a few different populations. So it was pretty quantitative, not focused on coloration at all. And one day, Caitlin said, "Beth, why are painted turtles painted?" And it really just got me. It was one of those things where I'd been working with them at that point for 2 years, and I hadn't yet at all thought about the color. And it took this artist to really give me this different perspective that totally shifted my trajectory. And I like to think I've also shifted hers quite a bit too. Her work is really nature focused. So just recognizing how valuable it is to have a diversity of thought in fields and to have these interdisciplinary collaborations and conversations has been really significant to me. And so I've sought through the rest of my career to help people and to be personally making connections with artists as much as possible, for mutual benefit. I think everyone benefits when you have different different ways of looking at things. And so I've been really fortunate to have some different ways to manifest that. I've been able to co-teach a class here at NEIU with a graphic designer, Lauren Miranda. We teach a class called Design in Nature that really just we have half biology students and half art students, and we do a biology lecture and then we do an art project inspired by that. And it's so fun to get to have these people who are usually in such different disciplines, thinking so differently, trained so differently. To have everyone work together to accomplish things is really valuable and important. And I've continued my collaborations with Caitlin also. We work together, and then have also been exploring some more graphic design, kind of art-focused work for science communications benefit. So a few different directions to incorporate art.
What's a book of fiction that has played an important part of your life?
Okay. Let me think. I'm a big reader, but I'm very much in spooky mode right now because it's October. So I've been reading a lot of ghost stories and horror, and I don't think those impact my research very much. so I'm trying to get my head away from those. Think about what are some things.
Was there a novel or something that stands out in particular? What would you say is your proudest accomplishment?
Ooh. My proudest accomplishment is the amount of research I've been able to do with undergraduates here. It feels so valuable to get to work with undergraduates. Like I benefit from it, but also I think that they benefit from getting to camp for the first time, getting to see turtles in the wild, for many of them for the first time. I've had students who've never left Chicago until doing fieldwork with me. And so every summer, just getting to share with them the beauty of what Illinois has to offer, what Wisconsin has to offer, how easy it can be to go camping and like break through that barrier is the thing I'm most proud of. That I get to do that every summer, take students and just change their lives means a lot to me. It's really, really powerful and really fun. And
yeah, definitely.
Every year I say, all right, this is the last, I'm going to take a break next year. I'm just going to do the bare minimum. And then I end up bringing a big group again. So
how many students do you usually bring?
So we're really fortunate here to have a student center for science engagement that funds summer research experiences. So I don't have to pay the students, the school will. And so we're capped at 4. So I usually bring 3 to 4 students, but now I've been partnering with other faculty and they've been sending their students also. So for instance, I partnered with a mathematician this year who also came to the field site and brought 4 of his students. And so we had a big crew of 8 students and a graduate student who were all working together to catch turtles. And it was so fun. Half of them were math students, and maybe not very outdoorsy. Half the biology students aren't always outdoorsy. And everyone's camping for the first time. Everyone's getting to experience what it's like to wake up to the dawn chorus and to catch turtles all day and collect data. And just the conversation, again, the interdisciplinary collaboration is super important to me. And so having that with a mathematician this time was really, really fun.
What are some of your hobbies outside of science?
Well, I have played roller derby for most of my science career. I actually just formally retired from it last year. And science can be a really lonely, even though it's very collaborative. It requires, at least in academic science, requires moving wherever the job is. And I struggled with that a lot. And roller derby was just the perfect way to have an automatic community, to meet people who are like-minded, who are athletic, who want to hang out and do cool things. And often there's a lot of other scientists in derby too. So that was a huge part of my identity for a long time, including when I've been here. But I just retired just because it was too much on my body. Ironically, I then broke my leg months later, but unrelated to roller derby.
Not doing roller derby.
First time I ever got this seriously injured. Ridiculous. But that was one of my big hobbies. Another hobby lately has become bird watching. So I'm a big birder now, partly because I was kind of shoehorned into teaching our ornithology class and local fauna class. But now I just realize how awesome it is that we, especially here in Chicago, I mean, we're in this migratory pathway. We get so many cool species, especially this time of year. And it's just really fun to get to try to identify all these colorful little quick moving warblers, for instance. So I really enjoy birding.
What do you think is the biggest threat to human survival?
To human survival? The biggest threat is humans. Easily. Yeah. are our own worst enemy and everyone else's. We're ruining everything about the environment, about ecosystems. We have such a we, as in the general public, has such a simplistic view of ecology and such a what's like a negative view of science that it's really gonna hurt us for decades to come right now where we are.
Why do you think so many people have a negative view of science?
I think a huge I think scientists are a huge problem, are a cause of that, not intentionally, of course. But the way that science is done, where we try to have this open transparency, we have these. It's so important that all the science we do is replicable, is detailed, that we produce as the main product of science these long, boring, dry papers, right? And that is the way that scientists need to communicate to other scientists. It's important to have all these details. It's important so that it's replicable and transparent. But when you're publishing only papers and that is the product of your science, nobody's understanding that. That is not effective communication. I don't have the solution. I don't think that any one person does for how to fix science and improve our view, but we need to change something, right? Every scientist knows that the way that we publish is broken anyways. There's all these, it's most of our science is taxpayer funded, but then our products are put behind paywalls, run by private entities, right? That's all a messed up system. So fixing scientific publishing, but also fixing and improving scientific outreach and communication is such an important part. And there are full-time jobs that are just, how do I translate this science to the people, right? And to some extent, every scientist has the responsibility to do that, but we also don't have the time, the funds, the training to necessarily do that. So not to blame the scientists entirely, because there's a lot of other bad actors at play, but I do think that the way that we do science is inherently gate-kept and makes it hard for others to access.
Because that is something you've been interested in, is science communication as well, right?
Yeah. I teach a science communication class here at NEIU. I designed it from scratch, and it's been in really high demand, and we're now considering making it a required class for our graduate program. And that's not to say that I'm an expert at science communication at all, but I find it incredibly important. And by teaching this class, I get to practice it too. I get to learn, how are the students using social media? How are they getting their science? How could we improve that connection? As well as getting to teach students how to do these things, how to translate their science into stories, how to turn their boring bar graph into something that people can understand, right? So it's kind of, there's just so many facets to science communication, and it needs to be something that every scientist is thinking about, but it's not often taught.
How do we combat the torrent of misinformation out there?
I don't know.
Yeah.
Yeah. I mean, we can try to flood the field with accurate information, but there's always going to be misinformation, and how people assess and how people, even educated people, know how to evaluate information is going to be constantly changing, especially in the next few years with AI and with all this, all the things that are happening. It's not going to get easier, is my answer. Yeah.
What advice would you have for a young upcoming biologist?
Yeah. The advice I'd give an up and coming biologist is follow your passion, no matter where it goes. Even if it feels really niche and obscure, just doing the thing that you love, or getting as close to the thing you love doing, is, I think, the best way to move forward. I think one of the more common problems I encounter with students is that they don't have a passion. Maybe they're just saying, well, I'm getting into science or biology because that's what my parents told me to do. I just want to make money. And I think that you can do that, and you can, I'm sure, find happiness in other assets of your life, and that's fine. But if you are passionate about something in biology, follow that for sure, right? That can lead you to exactly where you want. I'm not saying there aren't going to be hard times, but it's going to make those hard times worth it if you're doing something you love. I get paid to catch turtles in the summer. Like, that's so fun. I'm kayaking and catching turtles, and that's my job.
No doubt, right?
It's so cool. So, yeah.
What are some life lessons that have come to you through your research on painted turtles?
That's a good question. The big one, the first one that comes to mind, is that we don't know anything. That's what, to every question that we find an answer to, we're going to get 10 more questions, and probably that answer is not going to be straightforward. It's probably going to be way more nuanced. And so it's been really humbling to start off asking this really simple question: why are painted turtles painted? And 10 years later to still be trying to answer it. And you know, I'm it's not like I've been unproductive in that time. I've found answers, but not to that one what I thought would be a really simple question. And it's just been humbling to see this, that wow, even though this is a common, widespread, brightly colored species, we don't know why. We don't know why it's like that.
Yeah.
So that's been a big life lesson.
I think that is a misconception of a lot of the general public, right? Is that scientists know everything.
Right.
There's nothing left to discover, but
yeah, that's just not true, right?
Yeah.
For every single organism, on some level, if not many levels, there are going to be a lot of things that are unknown, right? And that's part of why it's fun to be a scientist, because you always get to be kind of widening this
yeah.
this you know, area of knowledge a little bit, even if it's an infinitesimal amount. You're improving the state of knowledge.
I agree. I think that is the excitement of science, right?
Yeah.
Is that the fact we're still making these discoveries.
Yeah, I think especially students who are in classes, in biology classes, they're getting so much information thrown at them. There's textbooks, there's scientific papers, there's lectures, there's all this information. It's really easy to think, oh my gosh, we've got it all figured out. So it's really when you just step a little bit to the right or left outside of what we know that you see, oh my gosh, there's actually way more that we don't know outside of that. So it's fun to help students realize how little we know. And it's fun to say, I don't know. I love when students ask me questions that I don't know the answer to. And it you know, that might be just because I personally don't know, but somebody does. But oftentimes it's, we don't know. Scientists don't know. Nobody's looked into that. You should look into it. You know, I get to say that a lot. So it's very fun.
Is there anything that you'd like to talk about or cover that I have not asked that you would like to?
We covered the art stuff. We covered the oh, I guess one thing I haven't talked about is this is a facet of biology and art and merging them, but I'm part of a network called the Biology Through Art Network, where it's more pedagogy focused. So how can we incorporate art and art assignments into biology classes to improve biology education and to improve learning outcomes? And that's been a really fun approach too, because generally I'm thinking about how do we combine like the practice of doing science with art, but also how can we combine the practice of learning science with art? And so through this network, we've been doing this kind of big controlled experiment across at universities across the US, where we do a art assignment, a creative writing assignment, and then just like a normal whatever assignment we typically would include. And we compare the learning outcomes. And that's been really fun. And I think regardless of what, even if we don't find that across the board the art assignments improve learning, I'm going to be keeping them in my classes. It's so much fun to force students outside of this box that they're in their biology classes. And I'm, at least in my classes, I am finding that it's improving learning outcomes. For instance, a lot of the classes I teach require identifying animals. And so just having students practice drawing a common grackle, a red-winged blackbird, and a brown-headed cowbird, and seeing these subtle shape differences really helps them learn identification in a way that I wouldn't have explored otherwise. And so it's really nice to get to bring art into teaching also.
Yeah. So you think they're becoming better biologists because of the art?
Absolutely. Yeah. And even if they don't, and students are often very afraid of this art assignment. I give them some basic art training, just like I don't consider myself an artist, but I can teach like shape, contour, some really basic things. I give them that level, teach them how to use the tools, and then set them loose. And they're not graded on how good their art is. The practice is really in paying attention to the shape, paying attention to the curves, these things that you, as biologists, we don't think about. We might think about a particular ornament or a name, but to make us see the world in a different way is really valuable and important. And similarly, we kind of added this creative writing treatment as like a control for like, is it actually the art that's improving or is it the time spent? But actually, I'm finding the creative writing is also similarly powerful. For instance, in my local fauna class, we do a creative writing assignment where they try to write a short story from the perspective of a fish, incorporating sensory information, for instance. And that's been really great too, because then you find the students Googling things like, do fishes see color? Can they sense polarized light? Do fish smell underwater? Like they're answering all these questions just to put themselves in this perspective of a fish for a creative writing assignment. And I think, again, just pushing people outside of the bounds of how we normally think is such a powerful tool. And for me too.
Do you think that improves their science writing too?
Probably. I don't know for sure. We haven't tested that, but that would be really fun to look at. I don't see why not, right? It's just more writing. Writing improves writing, so I
Yeah.
would think it would.
What do you think is the most controversial thing happening in your field of study currently?
Okay, in my field. I've been thinking about many other controversies right now, so it's nice to be able to focus on the field. I think this is not necessarily unique to color biology or to visual ecology, but I think everyone is kind of trying to figure out how to incorporate or how to deal with AI in our sciences. Um, AI can be a really useful tool for things like programming and coding, which a lot of scientists end up having to program a lot. But I think that this is going to be something that everyone's trying to deal with, is how do we use this as a tool that's effective and smart and not false, not misleading us? How can we incorporate it in a meaningful way? So I don't know if that's a great answer, but yeah, it's pretty broad.
Um,
A fun story about that paper, though. So this was a result of the work I was doing with painted turtles and realizing, like, oh, it's probably not the color that matters after all. It's this pigment physiology. How often are we ignoring what the underlying pigment does because we're seeing this bright, attractive coloration, when really maybe that's not what matters, it's the pigment that does. So I wrote this paper, or a version of this paper, with Julian Avery, who's on there, and submitted it when I was a postdoc. And at the time, we were writing it as, this is what we need to be considering, these other facets of color, because climate change is going to impact all of this, right? There's been a lot of work on how climate change is going to impact color, especially phenology-based color. Like, think about the snowshoe hares that change color with the snow. If the snow changes, but they don't, or vice versa, then it's going to be a problem, right? So there's these obvious, like, visual consequences of climate change, but also we talked about all these other things that color impacts, like thermoregulation, immune function, structural, like the actual like physiological structure of tissues. How are those things going to be also impacted by climate change in a way that might then impact the visual phenotypes? And we submitted it, and this was in probably 2017 or 2018, and it came back with reviews saying all of this is too speculatory. There's not evidence that climate change is going to impact these things. So then 3 years later, we had a chance to submit a paper again and did it. And by then, there were enough studies showing climate change is impacting these things. I was like, got him, but I wish this had come out sooner.
Right.
But it was one of those where it's just such a bummer that, like, yes, but the logic is all here. We have reason to believe climate change will impact every facet of a phenotype, of coloration as a phenotype, even if that color is not itself functionally significant. But it just hadn't been studied yet, and now it has a little bit. Still not very much, and so I'm happy that we have the paper out and that people can start thinking about the non-visual functions of color and of pigments. Yeah.
How has your work studying the evolution of biological diversity impacted your humanity?
Ooh, that's
a big one.
Big one. Let's see. So it's made me a better person in that it gave me another way to think about human diversity and the benefits of it, and this is not just in terms of phenotypic diversity, but also like the interdisciplinary collaborations I've done. All of these things have just helped me realize how much diversity strengthens everyone, right? In every way. And I don't mean diversity in any one axis, but literally just having a diversity of thought, of people, of everything makes everything better, even if it might be hard at first, right? Because we're being forced outside of our comfort zones. I think that that's still such an important part. So studying the evolution of diversity in organisms has impacted every part of my life because it makes me appreciate how much diversity there is in the world in every facet. It's kind of a cheesy answer, but I think that's the answer.
That's a good answer.
You queued me up for it, so it kind of had to. I'm not going to say it made me a worse person.
What would you like your legacy to be? If you don't want to answer, it's okay.
I don't know.
Uh,
I don't know. I guess my legacy as a scientist is not super important to me. What's important to me is my legacy as a mentor, as a teacher, as a researcher to undergraduates here at NEIU. I really do feel like I get to make a difference in student lives, and if I can be remembered for that, that would be a wonderful legacy. To helping students be less afraid of the outdoors, to help them get outside of Chicago, to help them appreciate wildlife. I love teaching our classes, local fauna classes here, because students come in thinking that we only have pigeons and rats, and then I pass them on the sidewalk and they're going, "Oh, a golden crowned kinglet," and things like that is just such a powerful, like, yes, you're never gonna see the world the same again,
yeah.
and that feels really powerful and
Changing lives.
yeah, yeah, and in minor ways, but I'm really happy about it, and
Yeah.
that legacy matters to me, I guess.
Where do you see your lab going in the next 5-10 years?
I have no idea, and that's part of the fun. I don't know what we'll be studying in 5 or 10 years. There was a while where I got really obsessed with cadaver dogs, and like how are dogs trained to detect human remains. And I still, maybe I'll go that way sometime. It's still kind of sensory ecology. It is sensory ecology, so it's still somewhat peripherally related. But I don't know. If I answer the painted turtle question, maybe I'll go, totally different into forensic science. I don't know, and that's the fun of it. I don't have to know. I get to do whatever. Yeah.
Well, Beth, it's
Yeah.
been a pleasure. Thank you for your time.
Thank you so much for having me, Paul. This has been really fun.